Hello everyone, this is Dr. Nasser and I would like to tell you about a unique red blood cell morphology known as stomatocyte. Stomatocytes are referred to as red blood cells in which their central pallor has narrowed down and developed in a form of a narrow slit or stoma, which means mouth. The reasons for stomatocytes formation are many. In general, the condition can be either hereditary or acquired. In the case of hereditary stomatocytosis, usually the problem is in a gene mutation that is coding for a protein to maintain the ionic balance across the red blood cell membrane. When there is a disturbance in this ionic balance, and then the water volume is not going to be able to control, and there is going to be an imbalance of water, and that causes the change in the morphology of the red blood cells. In the case of acquired stomatocytosis, it can be seen in various conditions, such as liver disease due to alcoholism. In this case, what we see here is red blood cells demonstrate about 30% stomatocytes. This feature is repeated across the field. You can see stomatocytes are present basically in every field. You can grade this about 3+. I think that would be a reasonable number. This smear demonstrates anisocytosis. Some cells are very small compared to some cells that are much larger. You do see Hal-Jolly bodies in a few cells in here, and Hal-Jolly bodies are going to be basically DNA remnants that is left from the time that the red blood cell was in the bone marrow. Hal-Jolly bodies are commonly seen in conditions such as splenectomy. If a patient has a splenectomy and remove the spleen, then you would see Hal-Jolly bodies in the field. And this appeared to be because of the number of these Hal-Jolly bodies that I see inside on this smear. I would suspect that this is the case. This person did not have a spleen, but you also can see Hal-Jolly bodies in megaloblastic anemia, in hemolytic disease, in thalassemia, even in myelodysplastic syndrome. However, in this case, I suspect that this is perhaps because there are too many of them, and this is due to splenectomy. This is not a Hal-Jolly body. This is a platelet that has fallen on top of a red blood cells. You make sure that be able to make this distinction. Another platelet in here. This is an artifact on this side, and that will conclude this smears analysis.